Welcome to MVIF Conversations, the podcast.
Welcome to another inspiring episode MVIF Conversation, the podcast. I'm Svetlana.
I'm Christina.
And we are thrilled to have a truly special guest today, someone whose work has fundamentally transformed how microbiome scientists analyze and visualize complex data sets.
Dr. Murat Eren, or as many call him Meren, is a computer scientist and microbial ecologist based at the Helmholtz Institute for Functional Marine Biodiversity in Germany. And, one of the core developers of anvi'o, one of the most influential open source platforms in modern microbial ecology and microbiology. He's authored or co-authored over 100 publications with nearly 20,000 citations, and has dedicated his career to making science more open, more accessible, and more reproducible. So, Meren, it's wonderful to have you here. I feel this is a very Balkan podcast because I'm Greek, Svetlana,
yes.
You are from Turkey. So.
Very true.
I would love to have you here. You've been a supporter of MVIF since its beginning, and we're grateful for your commitment to our community.
Thank you so much. Thanks a lot for considering me for this.
Okay, welcome, Meren. And one question from Serbia, from me, the first one. So let's start with your journey. You came to microbiology from a fascinating direction. So you studied computer engineering and cryptography in Turkey. So how did that path eventually lead you to microbes and microbiome science?
Yeah, that was quite a long journey, and it was mostly governed by serendipity. Basically, none of this was planned, so much so that I had never intended to become a scientist. I never thought about myself as a scientist. I moved to the United States in 2007 because of a relationship. And initially, I thought that I perhaps could continue my professional journey in life as a photographer in the United States, which did not work out because I did not know enough English at the time to pull it off, actually. Then I ended up in a PhD program because the PI, who was leading the program at the University of New Orleans studying nanopore biosensing, needed someone with good programming skills and had heard that one of the developers of the Linux distribution project in Turkey was now living in New Orleans without a job. And as a result of that, I started in his lab working on computer science problems like signal processing, machine learning, and so on. And then at the third year of my PhD, this professor who hired me for my programming skills fired me for my big mouth because I learned enough English during the time to be able to actually raise my concerns and so on. And at that time, I was 30 years old, and I still had no idea about microbes. I was not introduced to microbial life yet, but after I got fired, another professor, Michael Ferris, who actually worked with Dave Ward at the Montana State University to understand microbial life in Yellowstone National Park, he took me under his wings, and I learned a lot from Mike about microbes. He was at the time studying human microbiome, more or less. And then I started to feel like my computational skills could be more useful here to understand the past, the present, and future of microbial life, how they can be so small, so tiny, and still be able to deal with a lot of stuff that's happening around them. So that was my transition into microbial life, in a sense.
Wow, yeah, that's a fascinating story. So going back to the birth of anvi'o, your group is quite active in developing bioinformatics tools, and anvi'o is one of them, the most known ones, the most used ones. It was published back in 2015. So before anvi'o was available, there was a moment that you realized that the world was needing this tool, that there was a gap, let's say. Can you walk us through how anvi'o came to life?
Okay. As you will see at the end of this answer, is actually it's not going to be as fun as it could have been. The reality
as previous one.
There wasn't a moment in which I realized anvi'o was needed, actually. But what happened is the beginning of anvi'o was also completed thanks to serendipity, I would say. So during my PhD, I worked on like the last part of my PhD. I worked on 6NS ribosomal RNA amplicon-based surveys of microbial diversity. And at the time, the use of 97% OTUs, maybe the next generation doesn't even know about it anymore, but it was popular as a method to think about microbial diversity, even though they conceal tremendous amount of diversity within them. And so I took that as a challenge. The final product of my PhD was oligotyping. It was a computational approach that would report sequence variants from amplicon data at a single nucleotide resolution. I had developed it with the hope to dismantle the throne of 97% OTUs, because I really didn't appreciate what was happening to amplicon data at the time. Of course, I didn't know at the time that I was going to completely fail at that, in part because of MVIO. Luckily, Van Callahan, Susan Holmes, and others brought in the heavy machinery, and they managed to rob the king of its clothes finally. And I wasn't needed after all, unfortunately, clearly. But I still pat myself in the back, especially in when I feel very lonely, that I was among the first people to throw the stone at 97% OTs. But after my PhD, 2011, I started a postdoc with Mitch Sogin at the Marine Biological Laboratory in Lutzow. And Mitch at the time was the best thing that could have happened to me. He basically said, you work on whatever you want. The only problem was that I did not quite know what I wanted to work on at the time. But oligotyping was gaining popularity, and it felt like maybe it's a good cause. So I started developing another algorithm that was inspired by oligotyping, which I called at the time minimum entropy decomposition, which was basically automatizing the process of oligotyping, this incremental Shannon entropy decomposition that was relying upon to actually report proper amplicon sequence variants. And there was a lot of room for improvements in MED. I could see the light at the end of the tunnel, and I could see that this is going to lead to something useful. But the same year, I met Tom Dalmont, who was also a postdoc at the MBL, who had just started with Anton Post. So Tom completely brushed my ambition about 6NS ribosomal RNA gene amplicons aside, and he said, you know what, you should work on genomes or metagenomics instead. It was the end of my journey in the amplicon world. I mean, he basically made, kind of, not intentionally, but came across as like he was criticizing me for something I really enjoyed so much at this time. Okay, so at the time, genomes or metagenomics was relatively niche space in microbial ecology, in which Jill Banfield was the benevolent queen of the realm. She was the one who was really leading the charge for years of experience in that. And I admit that I had understood the challenge, but I honestly had not fully appreciated the power of genomes or metagenomics, or integrative opportunities it enabled at the time, or much deeper insights into microbial world we could have thanks to genomes or metagenomes if we had the kinds of software tools we needed in it, and so on. But nevertheless, Tom, myself, and Özcan Esen, who joined our effort later, worked on anvi'o day and night for the next 2 years, basically. Now, today I look back, anvi'o has over 200,000 lines of code. And it has grown quite a lot with new ideas from early career researchers, such as Eva Vesely, Sam Miller, Florian Trigaday, Evan Kiefel, Alon Shaver. A lot of people contribute to it. And it serves to a small but tight-knit community of like-minded researchers around the globe. It's not used a lot because in some ways anvi'o is going against the grain of the software development practices in the field. So it would have been very cool, of course, if I were to be able to say that there was a single moment in which I realized we needed this. But I am not that visionary, and there was no such moment in reality. I often think that we start doing things not because we know the need or clearly see the path to an end product, but we trust some sort of a vague feeling that makes it sound that it is worth our time. And in this particular instance, what led the emergence of anvi'o was the feeling that Tom was likely right about the future of integrated omics. And that's how anvi'o came to be, actually.
Yeah, true. And it's clear that the anvi'o is open source tool. So in general, what I would like to discuss with you is that open source software doesn't generate revenue, so it requires constant maintenance. And maintenance which many tool developers can't cover for a longer time. Some people come and go, and then of course you mentioned the team behind the MVIO. And how do you see a current open source effort in microbiome science? So what can we do better? So from your experience, let's say, what you can share with us.
Thanks for this question. I've been thinking a lot about these questions, and my opinion about these things have been shifting quite dramatically in the past couple of years, and I'll tell you why. I think microbiome science, like many other disciplines, has long suffered from the fact that a large fraction of software tools have been developed by good scientists who understand their systems with limited to no background or training in software development. And this created a long list of issues with serious implications, not only on the quality, accuracy, or reproducibility of our science, but also on what questions we can ask to our data and who gets to call the shots in our discipline. So these are fundamentally problematic, I think, things. But I also feel that these will soon be concerns of a familiar past. I think the large language models or artificial intelligence is so rapidly democratizing access to good software design principles. The cost of developing and maintaining high quality scientific software is dropping dramatically. So I think the best thing we can do right now is to start imagining that software is no longer the bottleneck and ask ourselves, what is it that we could be doing right now to take advantage of this new reality? I really think that we have entered the Star Trek era of computation. So tools like MVIO, where a group of people literally manually put 200,000 lines of code together, is in the past now. We are rapidly going into a new era. And I think we should use it to redirect our energy and funding toward those who are willing to do the hardcore microbiology part of our science, like whether cultivation, genetic editing, screening, functional discovery, imaging, you name it. So we actually can stop reanalyzing the same correlations with different data sets and start getting more mechanistic insights that can improve our biotechnology and biomedicine. There are growing societal challenges in our rapidly changing planet. I think we all recognize that. And I think we also recognize that microbiology is uniquely positioned to offer solutions for at least some of those societal challenges. And I think we will do better if we keep them on our radar. And this goes into resources we invest into software development. How do we actually contextualize software in microbial ecology? So that goes through my mind when I think about these things as of 2026, basically.
Yeah. So I was thinking, as you were mentioning all those things, that probably we need to shift. We need to use AI. You mentioned for the creation of the tools for the software, and to squeeze our minds for the better part of the outcomes and the analysis afterwards and what comes out. One of the main things that people have been using AI lately is to write documentation for tools, not only the actual code, but the documentation and the tutorials for it. But you have a very active community that has been working on, and you have very good documentation and people that have been actively supporting.
And in different languages.
Yeah. But how do you envision this going on in the future? How do you manage it? Because it requires time, and I know whatever thing requires time lately, it's really difficult for people to do.
And people.
That's a great question, because I don't think AI. We should use AI to write code as well as documentation. But of course, there has to be still a tremendous amount of input from us scientists. I am using it now, slowly trying to integrate it into my life. I'm from an older generation, so it's more difficult for me, but it's happening and happening very fast. Our group, we enjoy the idea of serving the community and doing it without cutting corners. So that's one of the principles on which we built our group in science, which immediately leads to the realization that the documentation and transparency of our science and tools is a primary responsibility on us. So that's how we feel. I'm doing my best, and I have been extremely lucky to work with those who feel the same about these things. But that said, without truly finding joy in such activities, no sense of responsibility could help anyone to dedicate so much time from their life just because of some principles.
Yeah.
And we do find joy in these activities, in part thanks to community feedback. That's what keeps us going, in my opinion. We're lucky in that sense, and I'm very thankful for everyone who ever reaches out to us to criticize us, endorse us, or simply ask questions, because that kind of feedback is what really propels us to continue doing this, in my opinion. And that will not change. I mean, I'm not going to be satisfied with AI feedback. I will want to know from real scientists how they see what we enable or prevent in their science to be accomplished.
Yeah, and I think the sense of community that you mentioned is really important. This is what we are, let's say, lacking these days overall.
And absolutely, I am one of the things that I really appreciate in this effort, MVIF. You are turning names behind papers into a people of a community. So that is, I think, by doing that, I know a lot of effort has gone into this, and behind the scenes, there are many more people who envisioned, found sponsors, and made it happen. The community, turning our field into a community, is an extremely important gap, and I think you guys are filling it very nicely. I always wanted to thank you for that, and I just grabbed the opportunity because this feels like a good moment to do that. And this
Thank you.
podcast
Thank
series also show your dedication to improve things, turn us into a community. So thank you for that.
Thank you.
Thank you. And now I would like to switch to really important part in not in only microbiome science, but science in general. So mentoring and relationship and support between juniors and seniors, and how to learn from each other, etc. So your commitment to better mentoring really stands out across your work, and first sharing freely knowledge through blogging, workshops by your team, and then when you conducted that survey on mentorship in life sciences. I'm really curious about that survey. So what findings surprised you the most
Thank you.
that survey?
Thank you for that question. So I think mentorship in science is one of the most critical roles senior members of our communities play. I experienced myself or witnessed such devastating outcomes of poor mentor-mentee relationships that part of me have always been curious to understand the sources of these issues and potential solutions for them, basically. The mentorship survey you refer to is, which people can find it online by simply searching on Google Voices of Mentees, was an attempt to improve that understanding on my part. And maybe translate some of that to others in case it's beneficial to them as well. The most surprising finding, there were many surprising findings actually, but the most surprising in my opinion was the realization that it was an extremely small number of mentors who ever asked for feedback from their mentees about their mentorship. So, this is, to me, it was very interesting because mentorship is a part of our jobs as senior people. We have to write grants, we have to get papers, we have to hire people, we have to mentor them and so on. And many of the things we do has an immediate consequence, a feedback loop that allows us to do better. We write grants, we get reviews, and we're like, okay, we didn't do so well, let's think more about it and so on. But when it comes to mentorship, there's no such mechanism. No one cares if our mentees are happy with us or not happy with us and so on. It doesn't even, I would say, unfortunately, influence hiring decisions of scientists. And so I had not realized until the survey that this was such a lacking aspect of mentor-mentee relationships. And I was one who never asked people I worked with whether the kind of mentorship they were receiving was the kind that they expected. So, I'm not saying that I am any better than the colleagues I am inadvertently criticizing here. And perhaps not so surprisingly, those mentees who worked with a mentor who asked for feedback at least once throughout their entire interaction period were substantially happier in the outcomes of the mentorship experience they had compared to the other group. So that was such a statistical gap between them. So I have this fundamental belief that human relations are complex, but not complicated. The more we communicate with the core belief that everyone is doing their best, the more complicated things can turn into complex things we actually can resolve. And what I just said is actually the job description of ourselves as scientists. We do take complicated things, we turn them into complex things with data, with analyses, and then we try to dissect them and understand them. And I think we can apply that to our professional relationships as well. But I also know that knowing these things don't solve anything. For instance, I think a lot about this person, but it has not been enough for me to be a good mentor for everyone I work with. Just like the way that I have not been a good scientist for every question I run into throughout my career. It's just, we have to accept these things and recognize them, I think. It's a constant battle, basically, being a good mentor, finding a good match, finding those matches that work so well together. If you were to ask me to summarize what I have learned in science since the moment I start my PhD about 20 years ago now, I would say that finding the right mentor is far more important than finding the right topic or right place or right institution for your science, because I think the implications of bad matches with the latter list of things is much easier to recover from than a bad mentor-mentee match. Part of me constantly mourns all the people we have failed in our discipline and either pushed them out or gave them lifelong memories that they don't want to remember. And it will continue to happen. But I think as scientists, we can take a bit of the control back and make things slightly better by trying to approach professional relationships the way we approach science. So that was seeing how easy it could be was the biggest surprise in general in that entire survey, because it really, when you look at the data, when you listen to people, it seems like most of the things happen due to some simple misunderstandings that are not resolved at the right time. Just like the way those simple misunderstandings at the beginning of our research would have significant implications at the end of it. So it's similar to that. So communication is very important, and being able to formalize it, being able to find the right context for communication is what we all are lacking a little bit, I think.
Yeah, fully agree. Communication is really the key. I mean, not only communication with mentors, but also between your colleagues, like communication between labs. Yeah. So not only within your lab. And what I saw from your activities lately is also that you open up for conversation out of your lab. So you created a new initiative, and people can reach out to you directly when they need mentoring support, and they are not in your lab. So can you please share with us the idea and how it's been so far?
Yes. After all the things I realized in science and heard the stories about mentor-mentee relationships, plus slowly reaching a level of maturity and finally recognizing or perhaps accepting my role in it, I wanted to kind of maximize my availability to those early career researchers who may be in need of a conversation with a senior scientist outside of their training environment, immediate training environment. And so the program, the mechanism you mentioned is one that I invest 1 hour each week for people who want to book meetings with me through an online interface, during which we can talk about anything, the difficulty of making long-term life decisions, struggles with funding, how to manage time and energy, how to deal with issues with supervisors, how to go over administrative hurdles, and so on and so forth. And imagine this is a session during which an early career researcher can vent about their struggles, and a senior scientist would do their best to share their 2 cents and feedback if, when appropriate. And so far, I had 10 meetings through this mechanism. And in some of those meetings, all I did was nodding, feeling completely useless apart from being a caring soundboard if I could. In others, I felt that I was actually useful to help people realize things that perhaps they did not consider before. This was an immensely enriching experience, at least for me. And I would implore my senior colleagues who may be listening to this right now to consider doing something similar. I know that there are many established scientists who would be more than happy to invest their time to help others. And I would be more than happy to help them with the technical infrastructure I came out with to make this happen with no additional cost, to schedule meetings with those who wish to meet me. Because that was the most difficult part of it, in my opinion. Like, how am I going to enable this to happen without having to take care of emails, find time in our... So I solved this, and there's a very simple solution I'm willing to share. So if you are a professor and you are listening to this, and if you feel like you are at that stage of your career where you can listen to early career researchers and their problems, to share your 2 cents thanks to your decades of experience in science, please feel free to write to me, and I will explain to you how I set this up, and I will help you set it up as well. That would be, I think, one way to, reduce the friction, how difficult it is for early career researchers to find their way, and who they are, where they fit, if their problems are real or common, or is it too specific, and so on. So sometimes all people need is just to chat with someone who seems to care, even if they don't contribute immediately to their problems and their solutions.
Yeah, and it can only take 1 hour of your time per week, but it can be very meaningful to the people that will actually choose to meet with you and choose to, to contact you and share.
So. I really, truly hope so.
Yeah. So you mentioned MVIF earlier in our community and our events. So taking, all this information about the mentor and mentee communication that you shared, how do you think that we can better facilitate having an honest dialogue in our events between mentor and mentees? Is there any way that we can, let's say, encourage mentees to speak up and mentors to be, as honest and
more open,
kind as possible? More open, yes, also.
Yeah, I think it is quite an admirable desire to facilitate more honest dialogue, but of course it's an extremely difficult one. And we don't get those nice things just because we want them. It's really difficult to achieve. I am probably underqualified to make suggestions here. There are actual social scientists who study these topics, and I really think we should maybe reach out to some of them and ask their opinion too. But the first thing that comes to my mind is perhaps pairing early career researchers with a designated conversation partner before the event. For instance, if there was a bit of additional time between the end of talks and start of Q&A sessions, let's say, this setup could allow pairs to exchange ideas and vet questions before the Q&A begins, and there could be learning experiences for both parties, whether it's an early career researcher or a professor and so on, whoever is willing to participate in this. I'm suggesting this because my experience in the Netherlands, I gave a few seminars in the Netherlands, and there is such a moment where you don't get any questions as a speaker, but people turn to one another and they start talking about the talk. So they start to make sense of what you said and so on. And it leads to much better questions at the end, much well-structured questions that now vetted by 2 people. So this is one way I can think that things could maybe this platform could envision. Another could be to create explicit spaces in the form of forums to do what I'm doing in this one-on-one settings that I create with individual early career researchers, but as a group where the power differential is acknowledged and it is not ignored, but it's a good thing, so that these spontaneous panels and spontaneous panelists of these forums can collect opinions on social aspect of science and hear one another and discuss and so on. Because at the end of the day, I think the more opportunities early career researchers have to interact with senior folk, the more easily they recognize how they fit into this complex network of history and ideas in their discipline. And perhaps more importantly, they identify the gaps they are uniquely positioned to fill and so on. So I'm not sure what you would think about the feasibility of such things, but these are the things that I can think of quickly.
Yeah, the thing is that from what we've seen so far, I think it takes a bit of time where you see people are willing to answer questions, they have things in their minds, but they want to see some, movement happening in the beginning. They want to see 1 or 2 questions popping up before they warmed up and they actually open up. And I think this is also what happens in conferences. You see people talking with each other because they have things to ask, but they want, confirmation that they're not going to ask something, stupid or whatever, although there are no stupid questions.
Yeah. GRC is an amazing mechanism. GRS, the early career researchers arrive at the conference a few days before. They have their own small symposium meeting where they create a network, a sense of belonging appears to emerge from those. And then throughout the conference, you feel they are more empowered to take the stand, say things, and ask questions and so on. We can learn from those things, I think.
Yeah, and thank you for suggestions. Yeah, for sure, we are gonna discuss them with our team. So, just one more question about mentoring. So, where does your passion for transparent, open mentoring come from? So, is it shaped by meaningful experience with your previous mentors, or is it something else?
So, I will be honest. It actually comes from selfishness. And let me try to explain why I say that.
Okay.
I am here as a product of many good mentors I run into throughout my life. From those who purchased school books for me so I could continue my education when I was in middle school, to those who forced me to take care of my mandatory internships so I could actually finish my undergraduate education, to those who made me read the lines I put down on a paper over and over again so I could start understanding what's wrong in them and could start writing better. I could name every single one of them, and I often remember these people very fondly. They are in me, basically. I live life with them and through them. But I also am here despite many bad mentors I ran into and survived. I could name every single one of them also, and unfortunately, I often remember them too. My passion honestly comes from the fact that I am too scared to be someone in someone else's life who will remember me for much longer than I deserve and for all the bad reasons. I very much would like to avoid this outcome for myself and for others as much as possible. So we all have better things to think about when we're taking a shower in the morning instead of people that, left behind significant dents in our lives. So I am selfish like that. And that's really where this is coming from. I am not an expert of any of these things. I am just actively thinking and trying to find some formal understanding of why and how things go haywire and lead to those horrendous outcomes that should never be tolerated. And I also have to say that I am not the best mentor I know. I do my best, but things sometimes don't work. And I unfortunately recognize it as well, which makes everything much more difficult. Even when you try very hard, that doesn't mean that things are going to work. Trying is not the insurance that you will achieve something. But trying is better than not trying, I think.
Yeah, for sure. So going back into the science part of your life.
Yeah, we can continue about mentoring for sure.
Yeah, I think we can talk about, everything forever. It doesn't really matter.
Yeah, especially about mentoring. Yeah.
Yeah, coming back to the science part. anvi'o is one of the tools that you have been a core developer and you have been involved in its creation. So how do you think the landscape looks now after so many years and after all these techniques and all these tools and all these pipelines in the field? What is the, let's say, the next thing? How are the tools, evolving?
There are, of course, a lot of tools. MVIO has, as I mentioned, a very specific philosophy behind it. And in some ways, it goes against the grain of current popular software development practices in life sciences. And we are the first ones to often advise potential MVIO users to consider other tools instead of MVIO. So we recognize the richness and the large number of options in our field. But I would like to say a few words about not where we are today, but where we are going to be in like 10 years, thinking more broadly. I think we can all agree that the software landscape is rapidly changing. And I think the pace of change will increase as we learn more and more how to employ large language models more effectively in our work. In fact, I will go one step further and suggest that if everything continues in the trajectory things have been going so far, I don't think there will be any software tool people will be able to name in our discipline as soon as a decade. Writing code, so bear with me. Writing code has been difficult, right? And it could be only done. It could be done only by a relatively small number of people. And software tools have been our means to make sure that the code written by those people could benefit those who did not know how to write code themselves or didn't have the time for it. But when everyone can actually write code well and rapidly, well, no one will need to run anyone else's code anymore. And we are slowly getting there. And I know this sounds like a syndrome from The Incredibles. I don't know if you remember the movie, who said when everyone is super, no one will be. He had this problem with, supers. I don't have a problem with coders and software developers, but I think this is the world that awaits us. And actually I welcome it, as it will empower the life scientists to make computers do what they want instead of what bioinformaticians think their computers should do for life scientists. Of course, infrastructure, standards, need for reproducibility will still matter, but I think the level of abstractions will change even if they don't fully disappear. And this will happen very, very fast. I am teaching a course at the University of Oldenburg called Programming for Life Scientists, in which we talk about LLMs a lot, the transformer architecture, and its scientific and societal implications. And it is perhaps in part because of that, when I think about the evolution of software in our discipline and beyond, these are the things that come to my mind first. So we, I think, can stop thinking about the future of software or difficulty of maintaining code or writing code, and start really thinking about the fact that writing code there will be a point it will no longer be a limiting factor. Then what do we do? Everyone can write code. LLMs are better programmers than anyone with the right kind of guidance and direction and feedback. So then what? Today there are a lot of great tools, but I'm afraid in 10 years, none of them will matter. We will not even remember them, including MVIO, I think.
Okay, could you please name those tools? Like you said, nowadays there are good tools. So yeah, my next question is actually that one. So could you please share with us so some of the emerging tools?
I can name tens, but
okay, start one.
But the problem is there are many more. So I would be leaving things out without intention, and I would feel very bad when I listen to this podcast again that, I forgot. But
okay.
Maybe we can find a middle ground. How about I tell you what excites me, not about tools themselves, but overall methodologies and emerging concepts in our discipline.
Yeah.
Because
I think probably in part what I was trying to say, that we need to focus on these things rather than individual tools. It speaks to that spirit as well. So I am excited about a lot of things, like many of my colleagues, obviously. For instance, one of them is the kinds of tools and ideas brewing in the processing and analysis of ancient DNA for ecological microbiome insights to synthesize geological timescales with the evolution of the enzymatic pool of life on Earth. Another thing I'm very excited about is molecular approaches that enable in situ community gene editing to better test some of the hypotheses that range from gene function to identifying currencies of microbial interactions and so on. I'm also excited a lot about new approaches in microfluidics and droplet chemistry that now enable high throughput cultivation experiments for us to gain insights into how and to what extent microbial consortia work together and under what conditions they break apart and so on. I'm also excited about the work that makes predicted protein structures more accessible to life scientists to contextualize the implications of sequence variants and chemical transformations of organic matter our proteins work with, and how all those come together and so on. I know this is such a cliche to say, but I really think that we're living in the golden era of microbiology. And I genuinely hope that we will also witness these advances to turn into not new billionaires, but positive societal outcomes. And I really am quite excited thinking about all these things, trying to constantly define my role, my place in this, but also hope that others will also join in this effort to think about what is really, if we were to forget about tools and computational tools and the difficulty of creating software that could help us analyze complex data, what are the questions then we are interested in? Like, what is it really we want to achieve with our science?
Yeah, that's true. That's
Sorry, that wasn't a very accurate response. It was more
no names of the tools, no other labs mentioning. Okay.
No, but this is
okay. Switch to another.
This is like
I'm
when you
very
ask,
politically correct somehow.
Yes, you are.
No,
Okay.
it's like when you ask actors talking about their next, their next play, their next show, and you tell them, they're like, I cannot tell names because I will forget I will forget them. And
Yeah, but it's true. I mean, it's a
no,
challenge.
but it's true. So can I ask, what would you recommend for a new, let's say, scientist to delve into microbiome science at this point? Would it be any of the things you mentioned earlier? And what would be if you were to restart and to start at this point, microbiology and microbiome science, what would you choose?
Okay, what would I suggest someone who is new to the field? This could be the moment where I go back to my real personality and drop the political correctness. So this is the answer I am now. I will recover. So first of all, I think different times present different opportunities, and those of us who are lucky enough to take advantage of them, knowingly or unknowingly, end up making it in one thread of professional life or skipping to the next one. So my experience back then and people's experience today are not quite comparable, I think. But also that difference does not matter that much. It would be very difficult to advise people knowing that perhaps it's all serendipity and perhaps there is no need to enter the ring for a fight against the universe of probabilities by making explicit plans and taking advice. So I often recommend people to not take any recommendations from anyone. That's usually my general recommendation. But if I were to offer one now, I would tell them to learn how to better listen, to and make sense of their own gut feelings first. And then I would advise them then that listen to advice from others, nod your head, smile in agreement, and then go ahead and do the opposite. Don't take advice from anyone, especially from those who are in a position
especially
to give
from Meren, huh?
especially from Meren. Because you have to believe that it's your turn now to run the world, and don't taint it with ideas or by following input from others who had their own run. Just be a rebel. Declare war against everything sensible and do great work.
Okay, we will cut out this part.
No, but this is exactly what I would recommend to anyone who are just getting into microbiome science or into anything for that matter. Just follow your gut feeling. Learn how to make sense of your own gut feeling. Advice from people, it's the worst thing you can do to yourself, following advice from others. Just listen to them, but go ahead and do what your gut tells you. That's my opinion.
Okay. So let's wrap up.
Except people in my group.
Yeah. So let's wrap up this conversation. No, I really like. I would like to hear now for like conclusion. Yeah. So where you are heading? So a few years ago, you moved to Germany and focused on biodiversity change and ecosystem function. So what's driving your vision besides rebelling, for the next chapter?
What's driving my vision besides rebelling? To be perfectly honest, at this stage, my vision is driven mostly by ideas of those who are better than I am, those who work with me, who are younger than I am, who are more in touch with their reality and have time to think deeply and so on. Because as I get older and older and I go more and more senior in my career, my time to really do those things effectively diminishes as a byproduct of this machinery that grinds you to a sand that doesn't form anything. But one of the things I really want to do, if I could, is to put the effort to understand the processes that drive the transformation of organic matter. How and under what conditions labile or recalcitrant molecules are produced, transformed, and eventually consumed or not by members of life. How can we bring together the ecology and evolution of chemical substrates on our planet with the ecology and evolution of its microbial constituents? In other words, what I'm saying is that I would love to try to get closer to understanding the overarching principles of biophysics and biochemistry that enable biotic and abiotic forms of complex matter to co-evolve, which is the ultimate integration of everything, of course, and everything that is out there and that matters. And I hope that this is going to be the next chapter of my science, but I'm looking forward to seeing where serendipity takes us.
Oh, me too. We are also looking forward to it. Okay, Meren, this conversation has been really meaningful. And thank you for sharing your time and insights with us. It was really my pleasure. Christina, you can conclude.
Yes.
Please.
So this has been MVIF Conversations, the podcast. Thank you all for tuning in. Our guest was Dr. Murat Eren, or Meren, from the Helmholtz Institute for Functional Marine Biodiversity in Germany. So until next time, stay curious, stay connected, and don't forget that microbes are everywhere. Thank you very much, Meren.
Thank you for listening to MVIF Conversations, the podcast. Until next time.